think, if I remember correctly, we stopped somewhere about here. Does that kind of jive with, no? Can we get further than that? It's about that. So I think, right, we were talking about pancreas, because we had gotten down to the small intestine, we got past the stomach, we got to the duodenum, and then we were talking about all the accessory organs that dump into the duodenum. So we talked about the liver and how it produces bile and what it looks like. We talked about the gallbladder, how it stores and releases the bile. And then we started talking about the pancreas itself. And we mentioned how, and we see this in lab today, how most of the pancreas is made up of our pancreatic acini. These have a digestive function. But we also have these lighter cells making up these pancreatic islets, which are endocrine in function, releasing things like insulin and glucagon. So these are the cells that are producing pancreatic juice, which helps to neutralize the acidic chyme coming from the stomach, and also a whole list of different enzymes. So these are the enzymes that we were finishing up with. We were saying that the pancreas, whereas previous to this we had seen, oh, this organ releases one enzyme, this organ comes along and releases one enzyme along the way. Now the pancreas, because we're starting with the small intestine where we really need to finish that digestion, we need enzymes for everything coming in here. So you can see proteolytic enzymes coming from the pancreas are digesting proteins and peptides, pancreatic amylase to complement our salivary amylase that we got earlier on and continue the digestion of carbohydrates like amylose, and then pancreatic lipase to complement our lingual lipase that we had seen previously to work on the digestion of fats. And remember, this is where we're going to get a lot more efficient digestion of fats because we're going to have a new lipase in combination with bile, our emulsifier, coming from the gallbladder and the liver. So we can really start breaking down our fats there. And then, technically, our nucleic acids are also some sort of, organic nutrient molecule that we need to break down. So we have nucleases to break those down into their subunits, which are going to be our nucleotides. Right? So this is something that we'll kind of touch on as well at the very end. We'll review this whole enzymatic or chemical digestion of our carbohydrates and our proteins and our lipids and our nucleic acids. So we'll go through that and remind ourselves of, okay, what are the enzymes that digest them and where do they come from? What are the substrates? What's the product? Things like that. So we need to know that chemical digestion very well because that's the whole idea of this system. Of course, we have big organs that are contributing to both chemical and physical digestion, but when it comes down to it, it's down at the cell and molecular level, right? We're releasing these chemicals, these enzymes that facilitate that chemical digestion where we're breaking chemical bonds. Okay. So we're kind of finishing up the pancreas then, saying that it releases pancreatic juice, which is alkaline, as well as those pancreatic enzymes. So how is it controlled or regulated? Well, there are two main hormones that come from the duodenum, because the duodenum is this mixing point for, we're getting new stuff to process from the stomach, and we're getting these secretions coming in from the accessory organs. So the duodenum has to be able to communicate with those different organs. It has to say, okay, stomach, I'm ready. Go ahead and send me a new load. Or, hey, stomach, slow it down. I'm still working on this. It has to communicate with the pancreas and the gallbladder and the liver to say, hey, I've got food here. It's time for you to give me your secretion so that we can start digesting it as well, right? Like we said, it's a bit like your washing machine or your dishwasher where there's a point in the cycle that, okay, we've wetted the food in there for a while now. Now we need to dump the detergent in there. So it's going to dump in at some time, and this is where it's going to happen, in the duodenum. This is where we're going to need all those secretions. And so this is where the duodenum can communicate. So secretin and CCK, which is cholecystokinin, is going to be secreted when we have these chemicals, these food nutrients that are arriving down in the duodenum. And so when we release them, the CCK is going to tell the pancreas to release its enzymes, and the secretin is going to tell the pancreas to release its sodium bicarbonate, which is that pancreatic juice, which is going to neutralize the acidic soup or chyme that's coming down from the stomach itself. Okay? So it's a good way for us to communicate via those hormones. And we also have some stimulation, some neurostimulation. So parasympathetic coming down through the vagus nerve. If you remember cranial nerves, we had 1 through 12. The vagus nerve is number 10, and it's one of the odd cranial nerves. It exists, comes out of the brain, but not only does it innervate, areas of maybe the head and neck, but it goes down into the chest as well as the abdomen. So the vagus nerve has parasympathetic stimulation that says, oh, okay, I know I've eaten these things as well, right? I've been smelling food, I've been tasting food, I've been swallowing food. So it's going to be coming to the pancreas soon, so we better start neurally stimulating the pancreas as well. Bless you. So this is just kind of a checklist of some of our major hormones, because one of the topics that we had at the beginning, which also counts as one of our exam objectives, is discuss and understand the control and coordination of the digestive system. And that's always going to be, okay, what's the neural control? The brain's going to be in charge. What's the endocrine or hormonal control? And then what's like the local control? We're going to see that when we get to, we saw it with the heart and the cardiovascular system. We're going to see that with the urinary system coming up as well. So for neural, remember we have autonomic nervous system, primarily rest and digest, parasympathetic fibers like we were just mentioning. And then we have these hormones that come in along the way too, that are secreted from different places along the way that influence, usually increase our activities, our digestive activities. So gastrin, right, gastro, that one comes from the stomach, and it increases the stomach activity, interestingly enough. So while it does have some far-reaching effects, it has more local effects. So we have gastrin, we have then secretin, and then there's the full spelling of our cholecystokinin. So these are coming from the duodenum and communicating with the pancreas. They can also communicate back to like the liver and the gallbladder to say, hey, you know what? We need your emulsifiers. We need your bile coming in at this point too, right? So the presence of food, and we actually have chemoreceptors which say, oh, we got proteins coming in, we got carbohydrates coming in, we've got fats coming in that we have to digest. Those chemoreceptors will then give these enteroendocrine cells the stimulus they need to say, oh, I need this type of secretion, I need this type of secretion, and I'm going to stimulate those organs that will release it. CCK is also one that is a feedback mechanism that goes all the way back up to the brain and contributes to satiation, right? So we have mechanoreceptors which say, hey, we're full. We have chemoreceptors that tell us what's in the food that we're eating, and we had enough of that, do we need more of that, etc. But also, we have these chemical feedbacks that can tell the brain started the process, right? Higher brain centers saying, oh, I'm hungry and I'm smelling things, etc. I'm thinking about food. And now we need a feedback mechanism to say, hey, we've gotten food, we're processing the food, we're probably good for the food. Stop putting more food in via ingestion, right? So CCK can go back to the brain as well, like the hypothalamus, and tell us to slow that down. Now this one, you don't have this in your slides because I just actually added it in, just because, as usual, there's always new information that we're learning about how the body works as well. This is another hormone that comes into play, glucagon-like peptide 1, GLP-1. This one comes into play when we get down into the ileum, the last section of the small intestine, and the large intestine. And for this one, I put it in here more for interest sake. So don't panic. It's not like we're like throwing extra things in that may be on the exam. We're not going to have a specific question about it. But if you got the question where you're talking about control of the digestive system and you wanted to list an example hormone, you certainly could use this one too. But you have your other ones that we were just talking about. In any case, the interesting thing about this one is it comes from the ileum. That's a new one, not duodenum. It comes from the colon. Oh, now we have another organ of the digestive system that is digestive, but also somehow part of the endocrine system, right? Releasing hormones. It's coming out from these what are called L cells. And this one has a lot of different feedback mechanisms, as you can see. But the same thing in terms of chemoreception is what gets this going, right? So if we're reaching the ileum or reaching like the first section of our large intestine, and we're having those digested nutrients, then we need to feed back to the other organs saying, okay, well, we've digested a lot of these organs now, we're absorbing them now, because that's what happens in the small intestine, and maybe as we get down to the large intestine, we're kind of finishing that up a little bit. So now it's like, okay, we can stop this whole process of digestion. So the digestive tract, we see it slows down the stomach activity because, oh, the intestines are working on this now. We don't need any new food for a while, right? So we only need to eat maybe a few times a day, and we start storing a lot of that energy. So some of the endocrine function from the pancreas, we start releasing insulin. The whole idea with insulin is that it causes our cells to store glucose inside certain organs, right? So rather than having this high glucose level, right, hyperglycemia, we can store it for later on. We can store it in the muscle cells, that's what you see down there, and we can store it in the liver. And then we're going to need insulin from the pancreas. That's the signal to those organs that tells them, hey, go grab that go grab that glucose that's in the blood and store it away for later on. So that when we need it hours down the road or more, and we haven't had anything to eat for a while, we can now start releasing that stored glucose instead, right? And then it also, you see how it has feedback? Feedback that leads to a decrease in appetite and things like that. So it's a feedback mechanism that says, yep, I got food that's being absorbed all throughout this. So we're good on nutrients for a while. So we can decrease the food intake, decrease the water intake, etc. You can see that there's some other effects as well that they're finding out in terms of secondary effects too. So effects on like bone, because we can store some of these nutrients in there, because often we take calcium in as well. Remember that. We'll talk about that next. You can see there's cardiovascular effects too. So the reason that I mentioned this too, not only is it an important hormone, but you may be hearing a lot about this drug recently, right? So Ozempic is one name brand of a new treatment, a new chemical that they're using. A chemical is called semaglutide that is a glucagon-like peptide agonist. So an agonist, if you remember back from muscle, there's antagonist, works against. And we'll see that even with some of these hormones, right? They work for and against. They work opposite to each other. So an antagonist is opposite. Agonist means that it works like it works with it, etc. And so with these drugs that people are taking, injecting, the idea is that they are using it for weight loss. And so people that are extremely overweight, right, to the obese level or something like that, where it's becoming a concern in terms of cardiovascular health and things like that, joint health, etc. Or they are type 2 diabetic as well, right? They're becoming insulin resistant, so their pancreas will release insulin, but those organs that are supposed to store glucose aren't receiving that message anymore. So by using this agonist, it'll utilize the same pathways to tell these other organs to do this. So for instance, it will tell the brain after you've had an injection, I'm not hungry, I'm not going to eat anymore, I'm good. That's part of the problem in terms of if we reach this imbalance where we have too many nutrients going in, we're not using them enough, so the body is storing them instead. So if we can decrease the input, the ingestion, and of course, in combination with exercise to burn some of that energy off, then you can get to more of a balance, right? So by injecting this drug, you are kind of stimulating this feedback mechanism that decreases energy input. It also can help to get some of those storage organs back to receiving the signal for insulin so that we can store glucose. So it helps to regulate glucose levels as well. And there is some, kind of cardiovascular benefits and things like that.
Lowers your cortisol levels too. Yeah. And so for night shift workers like me, I lost 85 pounds and I got like a whole new life. Yeah.
See? And up at the top there, if you look at the brain, there's a decrease in inflammation as well, right? Inflammation has to do with stress too. So inflammation happens all over the place as well. But so there's other perks as well. So it does have an effect on a variety of different organs that we see there. So, now culturally, there's the idea like, okay, so there are medical uses for it, that people have a need for it, etc. And then there are also more kind of like behavioral and cosmetic, like, well, who would want to lose an extra few pounds? And I do it with an injection, and I haven't been able to be very successful in, doing it by diet and exercise and whatever. So people are spending their own money to utilize it as well. But you always have to be aware that there can be secondary effects or unforeseen effects because we're adding it in there. It's kind of like if we think about other types of injections, now if you are diabetic and you're injecting insulin, that's because you're not producing insulin, right? So you're taking the place of it. If you're talking about, some people will use steroids for certain things. If we're talking about bodybuilding or something like that, by injecting steroids, you're seeing more of an effect, right? An anabolic, a building effect, but then you're taking the place of your normal secretions of that. So sometimes what you'll see is the body's like, oh, well, we're getting it from someplace else. I guess I don't have to handle that anymore. So there may be effects where, again, if your body is getting it exogenously, then it may release less of the GLP-1 instead. So they have to kind of monitor those things. And some people have had adverse effects too, where the organs that are affected, like their gallbladder and liver and things like that, can kind of become inflamed or something. But it's working for some people, and it's something that is, again, kind of a newer development. So, and it all has to do with that hormone that we see right there. Okay. So what we need to do is continue moving on, though. We had talked about pancreas and liver and gallbladder because we had gotten to our pathway, we'd gotten to the duodenum there. And we just want to remind ourselves that the duodenum is one of three sections of the small intestine. The first shortest section, where it's more of a mixing place, and it's really going to be the jejunum, the middle section, and the ileum, where that complete digestion is going to occur, where we're going to have the time and the space to allow that physical and chemical digestion to complete, where we can break these things down small enough to absorb them across the body wall. So what we're seeing is, by the time we get to the duodenum, we've had a little bit of chemical digestion of carbohydrates and proteins, minimal fat digestion like we saw maybe from some lingual lipase, but with those pancreatic enzymes, we're going to get a lot more. And in fact, there are more digestive enzymes built right into the wall of the small intestine. So as the food comes into the contact of the wall, like those villi and things like that, and microvilli, we have a bunch of embedded enzymes right there to make sure that we have complete digestion occurring. So we're releasing into the duodenum. In the duodenum, we're mixing with some of those secretions. And as we move through the small intestine then, we're going to finally get those nutrients down to be small enough to absorb them across the body wall. So virtually all of our nutrient absorption is occurring in that small intestine, as well as almost, or at least most of our water absorption and our electrolytes. So that's why we have such a long section there. That's why it has all that surface area with the plecus circularis, the circular folds, with the villi, with the microvilli. Because the more area we have, the more we can absorb across the wall there. Okay, so we're going to be squeezing it along and moving it through that small intestine. Again, our jejunum and our ileum are really there to complete the digestive process and absorb those things across. We'll talk about how they move carbohydrates across, how they move proteins across, and lipids and things like that coming up towards the end. But that's the role of our small intestine. So we have this almost complete digestion occurring in the small intestine. When we get to the large intestine, then what's going to be there are the leftovers, right? Meal remnants, things we couldn't digest or something like that. Bacteria possibly, mucosal cells that have been shed along the way, and debris. Oh, well, sometimes if you drop something, you dust it off, you're going to get some debris in there, or cellular debris or something like that. So that's what's moved into the large intestine itself. So the large intestine, we know, starts in the lower right-hand quadrant with our cecum. And then ascending, transverse, descending, sigmoid, S-shaped sigmoid colon, leading to our straightened rectum, then the anal canal, as well as the anus, the opening towards the outside there. We know that it has a unique shape to it, right? So it has these pouches along the way here that are called haustra. And we have these really strong longitudinal muscle bands, which are called the taenia coli. Again, lots of different spellings of that particular structure. We have our two flex points here, right? Our right colic flexure, also known as hepatic, and then our left colic flexure, also known as our splenic flexure. So this organ itself is going to be like a drying rack, right? Nutrient absorption has basically already occurred. So now we said we have these meal remnants entering here. So we can pull in a little bit of extra water, a little bit of electrolytes in there, and then some vitamins and things like that, but not a lot of nutrients per se. So as we mentioned, some unique anatomical features that we see there. These we hadn't seen before. Epiplaque appendages are connected fat. So we saw that in the sheep and even in the human cadaver, we see fat stored in different places. And those little bits of fat that we see here can even be along our large intestine. And as we get down to the very end of it, right, we're moving along, we're reabsorbing more water, so now it's becoming semi-solid again. So that's why we have to have that stronger muscle to move it along. As we get towards the outside, right, we're going to need to finish propulsion and absorption and secretion, and we're going to need to be able to accomplish defecation. So as we get towards the anus, we've got muscle bands, again, like we've seen along the way, right? Sphincters, muscle bands. But in this case, we have two sets of muscle bands. Mostly what we've seen is smooth muscle, and we do have an internal sphincter of smooth muscle. But there's also an external sphincter that is skeletal muscle. So we have involuntary backed up by voluntary. So that way, right, normally as the food is moving through, it has this pace to it, this natural pace that's coordinated. And so once our undigested meal remnants are reaching the last sections of our alimentary canal, the natural response would be, okay, it's there, let's let it go. Like as soon as it arrives there, let's let it go, right? But that's not very convenient for social situations. So it's good that we have skeletal muscle there that can have voluntary control saying, oh, wow, I can feel those stretch receptors going off and things like that. So I'm going to have to start planning when's going to be a convenient time to let that go. So that's why we have that skeletal muscle there as well. So we've got a lot of mucus secreting cells along the end there. We have the sinuses that are going to be compressing the feces as well as it's entering those last segments. And a lot of blood vessels all along the way for absorbing things. Inflammation of some of those blood vessels, particularly veins in those last sections, are what we call hemorrhoids. So when they become very distended instead. Now, another unique thing about the large intestine is that the large intestine is kind of populated with a large population of and variety of bacteria. So in this case, we have bacteria that are populating the large intestine that either have survived going through this whole pathway or they've actually entered from the outside, right? Because as we get to the outside, we're exposed to things from the outside as well. And the bacterial flora that we have are largely beneficial. So these are beneficial, or you might say even symbiotic, bacteria that live within us. And so they colonize the colon. You're seeing colon twice there. We give them a home, if you will, and they can benefit us in a few different ways. So for instance, they can break down certain things that we can't. We know that chemical breakdown of food items takes the right enzyme that fits into the substrate just perfectly. And we know that there are certain things that we can't digest. So for instance, fiber, etc. And so fiber helps to move things along through our digestive system, but certain plant fibers our bacterial enzymes can break down, but we can't. So they may break them down with their enzymes and fermentation. And so that's why you may get gas associated with breaking down certain plant fibers, etc. So that flatus is basically gas that we get in the colon from these bacterial activities. But when they do that, though, we can get extra nutrients, a small amount of nutrients like vitamins that can be produced and then brought into the body for its uses. Vitamin K is associated, in fact, with blood cell production. So that comes from bacteria. Now, in addition, with these bacteria, the good thing about these bacteria is that, like we said, they either don't hurt us or they have a beneficial function. And in fact, there are so many of them. There are more bacterial cells that you're carrying that are not you compared to the cells that make up you. So they are very, very numerous in there, and each person is a little bit different in terms of the diversity of these bacterial species that they carry. That has to do with the food you eat, where you live, and things like that, as well as kind of what your body will tolerate, too. So it's a little bit different between different people. And they're, in addition to these positive aspects of them, in addition, they help to keep out our pathogenic bacteria. So disease-causing bacteria will want to inhabit this neighborhood as well. And if you have a healthy set of, or kind of complement of beneficial bacteria, then our disease-causing bacteria don't have a place to start to thrive, right? So that's why if you've had some sort of issue where you had to take antibiotics and you got rid of your beneficial bacteria, sometimes people will get some sort of infection, right, from other types of disease-causing agents like C. difficile, etc., that will take up residence where our beneficial bacteria ended up kind of evacuating instead. So it's important to get them back. That's why they talk about eating yogurt afterwards. When you talk about active cultures found in things like that, those are bacteria. And so those are beneficial bacteria that you're reintroducing to the system when you've had a problem, you've got rid of them, like diarrhea, or you've gotten, like I said, antibiotics or something like that. Now, the funny thing about it is that, again, with newer research, they're discovering more and more benefits. Of course, we're looking at digestive and immune function, things like that. But what they found is that there are a lot of different connections between the bacteria that we have in our gut and the rest of the body. So some of their secretions and metabolic processes will get into the bloodstream, right? Because they're released and picked up. And they can affect lots of different things, like lots of our internal organs, and even feed back to our brain and can affect our moods as well. So looking at gut bacteria and having healthy gut bacteria is becoming more and more important. It's like we're seeing these microbial influences in our body that almost like they are the ones controlling us that we really had no idea about. And we're kind of making changes and making accommodations for them as well. So it's kind of interesting. So this is just talking about, again, if you lose those beneficial bacteria, different types of pathogenic bacteria that may take up residence and cause things like colitis, right? So colitis is, there's our itis again, inflammation of the colon. And in fact, one of the newer treatments for if you get one of these antibiotic-resistant pathogenic bacteria like C. difficile is they will do fecal transplants now. They will take fecal samples, right, poop samples from somebody with healthy bacteria in their colon and transfer that to somebody else, and then those bacteria, those beneficial bacteria, will repopulate the large intestine of the person who is ill. Now, it sounds like something where you're like, ew, but at the same time, right, if you contract one of these antibiotic-resistant bacteria, right, if your body can't get rid of it and antibiotics can't get rid of it, then what's it going to do? It's just going to continue to spread, right? So people die from that. So it's the treatment is relatively simple. It's something that they are starting to apply more and more, and it really takes advantage of these beneficial bacteria like we were talking about. Okay, so as we were talking about, as we get down to the large intestine then, it's basically like a drying rack. We're absorbing the last little bits of water. Do we need it or not? Vitamins, electrolytes, and things like that are going to be pulled in via the large intestine. But for the most part, our nutrient digestion is done. So we're going to be moving it through there, these meal remnants, and we're going to be contracting it along the way, moving it through the different haustra. And the activity that we get in the large intestine is still going to be coordinated with the other systems, right? So there is what's called a gastrocolic reflex. So that is stomach and colon. So it's kind of towards the beginning and the end. And it's good to know if we're talking about that whole disassembly line, are we about done with things? Are we ready for more things, etc.? So that's a communication pathway that includes even the beginning and the end of this as well. So in fact, as things are moving through the large intestine, peristaltic waves and things like that, it can lead to influences between them, right? So if you are taking more food in, that may hasten getting food out of there because we know that someone's going to be coming along the way, so we need to move things out of the colon to make room for what you're going to be processing. Likewise, the colic reflex, if you are getting rid of things, if you're defecating, then that kind of can increase the activity in our other sections, in the small intestine and in the stomach, etc. Okay, so for the most part then, that defecation, like we were saying, is going to be coordinated by muscular contractions, compaction, if you will, down in the last segments. stretch receptors are going to be coordinating that with the brain, and then again, vagal stimulation there, like peristaltic stimulation from smooth muscle, is going to allow that defecation to occur. So we have to relax the skeletal muscle, the voluntary muscle, and then the smooth muscle takes over. So you can see some of those feedback loops from our conscious control as well as our parasympathetic, our rest and digest control of our smooth internal sphincter as well. Okay. So what we've done is we've moved all the way through. We've done our kind of tracing of a food molecule through the pathway. We've talked about the different organs of the digestive system. And so now what we want to do is review, just focus on that kind of chemical digestion. The different nutrient molecules, what are the enzymes that work to digest them, where do they come from, and then what are our products and how do we absorb them across that intestinal wall. Okay? Because that really explains the process itself. Because these are the things that we're targeting. That's the whole reason that this digestive system exists, is so that we can get these nutrients into the body. All right, so carbohydrates. So carbohydrates are going to be things like our starches, etc. Complex carbohydrates are going to be our bigger ones, right? Our polymers, like we were talking about, our substrate. Those are the terms that we use for these larger molecules. So we're going to be starting with our complex carbohydrates, like that, like starches, potatoes, pasta, etc. And we're going to be breaking them down into simple sugars, which are also known as, right, these are monosaccharides, and disaccharides as well, bless you. And we started out with polysaccharides. Many of these units stuck together. So outside of physical digestion, chemical digestion is going to occur, and these are the enzymes that are used. We saw salivary amylase, we saw pancreatic amylase, and then when I list brush border enzymes here, the microvilli that we have down in the small intestine, right? We have circular folds, villi, and then microvilli along the apical surface of those simple columnar cells. Those microvilli, somebody thought they looked like a hairbrush, okay? So that's called the brush border. And as I mentioned, there are enzymes that are embedded within those columnar cells that will do the last step of digestion before we absorb things across the body wall. So we've got enzymes in all these places that work to break down those polysaccharides into disaccharides. And so when we're absorbing them, they are absorbed via a facilitated diffusion. So it's going to be high concentration in the lumen of the small intestine, right? Because that's where you ate all of these things. And then it's going to be low concentration across the body wall, inside of the epithelial cells, etc. So we're going to transport them. Oftentimes, these things are a little bit big, even as monosaccharides and things like that. Glucose is a little bit big. It can't just slip across on its own. So we need a facilitator. We need a transport molecule. So we often have a facilitated co-transport. It comes across glucose and things like that often come across with sodium. So it's a sodium glucose transporter molecule. So we transport it across, get it into the bloodstream, and usually then we're going to finish processing these in like the liver, right? The liver can do some storage of glucose, etc., as glycogen that we'll talk about later on as well. Okay. So that was carbohydrates, where we were talking about polysaccharides and getting down to monosaccharides. Now we're starting with proteins. That's the complex substrate, right? The polymer. Those go down to our peptides, shorter chains, less complex. And that goes down to what's the second term there? Amino acids, right? So those are going to be our monomers. Those are going to be our products that we see there. So for that, in terms of the enzymes, we have a variety of pancreatic enzymes. These are the collectively proteolytic enzymes. So trypsin, chymotrypsin, carboxypeptidase, those are proteolytic enzymes that digest proteins. We also have brush border enzymes, you can see again, that are breaking down those peptide bonds and making them smaller. So those are all for our proteins. The one thing we don't have listed there, what are we missing there? Another enzyme that digests proteins. Comes in the stomach. Starts with a P. So pepsin is the activated form. The inactivated form is pepsinogen, and that comes from the stomach. Right? Comes from, when we look at it, some of those cells in the gastric glands there. So those are all, oh, there it is. Sorry. It's up the top there. I missed it. So, and pepsin coming from the stomach, and then those other ones are coming from the small intestine here. We'll erase this. Ooh, see. Okay. So what we're seeing there is the digestion of those. We can see it going from a peptide here down to dipeptides. That's what we're seeing here, as well as our individual amino acids. We're going to do some facilitated co-transport to get them in there, facilitated diffusion to get them into our epithelial cells. Look, there's our microvilli, our brush border right there. Transport it through the cell and put it into the capillaries, right? We saw capillaries in the villi, stuff like that. So that's how we're transporting our protein products, our amino acids across like that. Okay? So facilitated diffusion generally is what we're bringing them across as. All right, now fats. Fats or lipids, remember, they're a little bit more complex. We saw this in our PhysioX exercise where we need two different components. We not only need an enzyme, but we need our bile as our emulsifier. Okay, so enzymes or chemical used are going to be bile salts from the gallbladder and the liver, as well as lipase coming in different places. Our main one is pancreatic lipase, but then there is some of that lingual lipase as well that helps to digest our different fats. But we've got to have the bile salts to do emulsification to help our enzymes chemically digest those fats. All right, so then when we're bringing them in is a little bit more complex as well. Let me show you an image for how these are brought in. These are our products. Remember, one of our main examples we were using before was triglycerides. That would be our larger form, our substrate, and that's going to give rise to like our monoglycerides and our fatty acids, right, our fatty acid chains. So those are going to be our products that we see right there. So this is just showing us some of that process. There's our large fat globule. We can see our bile salts attaching to it and breaking it into smaller fat droplets in a process that's called emulsification. So now we have way more surface area for our enzymes to be able to get in there and do their job of eating away at and breaking down those chemical bonds that we see there. Now, when we break that down, what we often get to is we get to these little organizational units that are called micelles. So rather than having just fatty acids, some fatty acids come across on their own, but oftentimes these slippery digested fat products are kind of easily slipping into other areas, right? Because lipid bilayers are also hydrophobic. And so we kind of package them into these little units, these little bags that are called micelles. So micelles are going to, kind of like fat droplets, gather them together and bring them to that brush border and release them, bless you, into the cell. So micelles are part of the transport process that are bringing the digested fat components to that epithelial cell. Then once they're brought in, they are packaged again. We see this packaging. This is smooth ER. Remember, smooth ER has to do with lipid, right, as opposed to rough ER. And so we see a packaging again of these products into another package, which is called a chylomicron. So a chylomicron is how it is transported inside of the cell after the Golgi apparatus and routed to the basal membrane where it can be released. And so we see those chylomicrons being released there. And notice that the chylomicron itself is a package, another type of package of these digested fats like fatty acids that are then brought into that lacteal. Okay? So it kind of brings it full circle when we were talking about the lymphatic system. One of the three key aspects and functions of the lymphatic system is to transport absorbed fats, because our lymphatic capillaries are a bit leakier compared to cardiovascular, and they can take in these larger chylomicrons for transport to places like the liver for further processing, etc. Okay? So we see our lacteal finally coming into play, that lacteal that we saw in our intestinal wall model, etc. So the digestion of fats is a little bit more complex. We need lipase, and we need the bile salts to help break them down. When we're transporting them across the membranes, we usually have these micelles that are bringing them to the epithelial lining of the intestine, and then chylomicrons that are allowing them to exit and be pushed into our lacteals instead. Okay? So all of our other nutrients, we're seeing them maybe go straight into the bloodstream, into the capillaries. And in this case, we get a little bit of that, but a lot of it gets transported into the lymphatics instead. Okay, and then finally, we have our nucleic acids. And our nucleic acids, remember we had ribonucleases. These are coming in from the pancreas. And these are breaking down nucleic acids down to nucleotides, right? Those are going to be the individual monomers from the polymer that we had before, right? Polymer or substrate, monomer, or product that we see over here. And these are going to be actively transported instead. So these are going to be pushed across. We know that we've got nucleotides inside of the cell. When we're doing our synthesis of DNA and RNA and things like that, we're going to be pulling them together. So there is some inside the cell. So we have to kind of push them across via active transport instead, because there's not quite the same concentration gradient that we saw with some of our other nutrients. Okay? Okay, so we saw that with our 4 nutrient molecules, our carbohydrates, our proteins, our lipids, and then finally our nucleic acids. For electrolytes, remember electrolytes in water and vitamins are also key coming into the body. So electrolytes, remember, are going to be charged particles or different types of salts and ions coming across. We won't get into a lot of detail about this because we're going to get into it as we do that chapter where we're doing fluid and electrolyte balance. But you can see how there are very important ones that are going to be coming across. Sodium. Sodium gets coupled with the absorption of glucose as well as amino acid, right? Facilitated transport. We get sodium coming in, potassium comes in, calcium comes in, right? All of these are very key for a lot of different tissues. Muscle tissue uses it in terms of action potentials, as well as getting the muscle to contract. Our neurons use them and things like that. So we've got to make sure that we have a lot of different electrolytes coming in from what we're taking in as well. Water absorption. Water absorption is occurring in the small intestines, like we mentioned, 95% of it. And this is via osmosis. Water always moves across a membrane or moves according to its own concentration gradient. So it moves from high to low water concentration via osmosis. So most of that in the small intestine, and then a little bit of it in the large intestine as well, that final 5%. It's kind of optional. Do our water balance is good? Should we let that go? Or if we're really dehydrated, right, we're going to reabsorb a lot of that from the remaining meal remnants. And so that's when our fecal material gets very compacted, right? You get constipated because we're having to reabsorb more of that water from the fecal material because you're not having enough water intake. And in essence, that's the story of the digestive system, right? So on one level, it seems like, oh, it's just a big pathway and follow it along. But we are realizing that there's a lot of different organs that each have a role to play, and not only on a macroscopic scale, but on a microscopic scale as well, right? So we have to understand those enzymes all the way down as well. Okay? Okay, so questions on digestive system. Understanding the organs, what each of the organs do, understanding the enzymes, where they come from, what they digest, how we absorb those across the body wall, etc. Yeah?
Does the large intestine secrete anything other than, I'm not counting the duodenum, but beyond the duodenum, does it secrete anything other than mucus?
Mucus is the main thing, for sure. Of course, we were talking about today where GLP-1 gets secreted there. That hormone gets secreted in the colon too. Yeah, so it gets secreted in the last section of the small intestine, the ileum, as well as like the first section of the large intestine. So, but mucus is a fine one, right? If you're listing all of your 7 different digestive processes, you got to define them and say where it occurs along the way. Definitely The motility of the propulsion occurs along the way, physical digestion along the way, as well as that secretion, because it's a mucus line pathway. So that secretion occurs everywhere along the way, too. Questions, digestive system? Yeah.
So you're talking about the gases that are created in the colon, and
Yeah.
that's only in the colon. No small intestines will be having like the.
Yeah, not from bacterial digestion. Certainly, it is possible that you can get, some gases forming even in the stomach and things like that, right? We swallow air or you're drinking carbonated things, right? You can have gases as well as fluids and solids down there. And then if you don't eructate, if you don't burp them out, now through the physical processes, they're going to continue on down the way, and that might distend the small intestine and kind of give you some kind of discomfort there as well. But in terms of generation of gases, it largely comes from the fermenting activities of those bacteria. Yeah. Okay. Then what we'll do there is we're going to stop.